welcome to Science Era. In this video, we're going to discuss complement pathways. The complement system. Complement system refers to the series of proteins circulating in blood and bathing the fluids surrounding tissues. The proteins circulate in an inactive form, but when activated, they act in concert in an orderly sequence to exert their biological effect. The term complement refers to the ability of these proteins to complement, augment the effects of other components of immune system, for example, antibody. Opsonization. The intrinsic ability of a phagocyte to bind to a microorganism is enhanced if the microorganism elicits the formation of antibodies that act as a bridge to attach the microorganism to Fc receptor on phagocytic cell. If the microorganism has activated complement C3b, the degree of binding is further enhanced by the C3b receptor. If both antibody and C3b opsonize, binding is greatly enhanced. Let's look at the complement activation outcomes. Three main outcomes are there: lysis of the cells such as bacteria, allograft, and tumor cells, generation of mediators that participate in inflammation and attract neutrophils, and lastly, opsonization, enhancement of phagocytosis. Advantages of complement activation. The first one is it triggers inflammation, chemotactically attract phagocytes to the infection site, promote attachment of antigen to phagocytes, enhance attachment or opsonization, cause lysis of gram-negative bacteria and human cells displaying foreign epitopes. Plays a role in the activation of naive B lymphocytes, remove harmful immune complexes. Complement system components. There are more than 20 components of which followings are the important: C1, C2 up to C9, factor B, factor D, factor H, and factor I, and properdin, C1 inhibitor, C4 binding protein, and S proteins are the main components of complement system. There are three type of pathways or three pathways which make up the complement system, which are classical pathways, menin binding lectin pathway, or alternative pathway. Let's look at the classical pathway first. This pathway involves complement components C1, C2, and C4. The pathway is triggered by antibody-antigen complexes binding to C1, which itself has three subcomponents: C1q, C1r, and C1s. The pathway forms a C3 convertase C4b2a, which splits C3 into two fragments. The large fragment C3b and can covalently attach to the surface of microbial pathogens and opsonize them. The small fragment C3a activate mast cells, causing release of vasoactive mediator such as histamine. Next is alternative pathway. This pathway involves various factors B, D, H, I, which interact with each other and with C3b to form C3 convertase. C3b BB that can activate more C3, hence the pathway is sometimes called the amplification loop. Activation of the loop is promoted in the presence of bacterial and fungal cell wall, but is inhibited by molecules on the surface of normal mammalian cells. Mannose binding lectin pathway is activated by binding of mannose binding lectin to mannose residues on the pathogen surface. This in turn activates the MBL associated serine protease MASP1 and MASP2, which activates C4 and C2 to form the C3 convertase, which is C4b2A. Lactic pathway. This pathway is initiated by splitting of C5 and attachment of C5b to a target. C6, C7, C8, and C9 unite with C5b, and this membrane attack complex, when inserted into outer membrane of some bacteria, can contribute to their death by lysis. Red cells which have antibody bound to cell surface can also activate the classical and lytic pathways and become susceptible to lysis. Let's look at some of the proteins used in complementary regulatory system. C1 inhibitor binds to C1, thereby preventing it from initiating complement activation. Properdin stabilizes the alternative pathway C3 convertase. CFF binding protein and decay accelerating factor accelerate the dissociation of C3 convertases. Factor 1 cleaves and inactivates the C3b and CFFb. Anaphylactoxin inhibitor proteolytically cleaves anaphylactoxin. CD59FIMIRL membrane inhibitor of reactive lysis inhibits MAC formation. Homologous restriction factor inhibits MAC formation, and S protein, also known as vitronectin, inhibits MAC formation. Complement activity in host. Complement also affects the innate immune system. Innate immune effects include lysis of bacteria, opsonization of microorganisms, and induced inflammation. Complement also affects the adaptive immune system. Adaptive immune system effects include augment or increase of adaptive immune response, enhance immunological memory, and enhance the antigen present. Then we have disease associated with complement system. The first one is deficiency of C5, C8, and MBL enhances the susceptibility to Neisseria infection. Deficiency of C3, severe recurrent phyogenic and respiratory tract infection. Inherited deficiency of C1 esterase inhibitor cause angioedema. Acquired deficiency of decay accelerating factor. Paroxysmal nocturnal hemoglobinuria. Mismatched ABO transfusion cause hemolysis and shock. Immune complex mediated disease include glomerulonephritis. This brings us to the end of complement system. Thank you for watching. If you like my video, don't forget to subscribe for more.